Hello everyone, welcome back. This is the February 15th lecture, part 1. Last time we were talking about the autorhythmic cells and how they generate action potentials. And we went through the steps of this. These notes are from last time. And I pointed out that the trigger for the action potential is when the cell membrane potential reaches minus 40, and that the slow gradual rise from minus 60 up to minus 40 is called the pacemaker potential, or slow depolarization. I just want to reiterate, why do we call that the pacemaker potential? Because the slope of that portion of this graph dictates how quickly we're going to get to threshold, and therefore dictates how quickly an action potential takes place. And because the SA node has the highest slope, or the greatest, most positive slope for the pacemaker potential, the SA node is the one that reaches threshold first and thereby sets the pace for the rest of the heart, because it generates its action potentials that then spread by gap junctions through all the other cells to cause the other cells to get to threshold before they would have on their own, because of slower rates of depolarization in those other cells. Now, take a look at slide number 23 in your PowerPoint. It shows this same graph, perhaps a little bit neater than I drew it, but you can see it's labeled pacemaker potential there. And later in this unit, we'll see how we can adjust the slope of that pacemaker potential in ways to get to threshold faster or slower. And we will find out that that's made possible by the autonomic nervous system, which is going to be able to adjust our heart rate to go up or down, mainly by adjusting the slope of that pacemaker potential. Now, I've mentioned a few times in this course that you guys have some really good online resources that came with the purchase of your textbook package, the Mastering A&P. And so what I want to show you next is an example of what I think is the coolest part of that. There's a whole bunch of learning resources in there, but the coolest part is the interactive physiology. So I want to just show you this clip and show you how to find it in your resources. I sent you a PDF document of how to register to access these materials, so be sure to take a look at that, register in. It has the course ID and everything. And I wanted to show you an example of an interactive physiology video that specifically addresses these autorhythmic cells. So here it is. So once you get into the online resources, the Mastering A&P page shows up, and you're going to want to click on Study Area and Launch Study Area. Take a couple seconds, you'll recognize possibly our book there, and there are all sorts of resources available here. But specifically the one I've been harping on is Interactive Physiology, the IP one. And if you click on that, every single unit will show up. I've already opened the cardiovascular system. So to give you an example of something we've been talking about, I'm going to click on action potentials in autorhythmic cells, and you can see what that looks like. So we'll click on play, and you can get an idea of what one of these looks like.
Cardiac autorhythmic cells in the intrinsic conduction system generate action potentials that spread in waves to all the cardiac contractile cells. This action causes a coordinated heart contraction. Of all the cells in the body, only heart cells are able to contract on their own without stimulation from the nervous system. In this magnified view of the heart, we see an autorhythmic cell adjacent to several cardiac contractile cells. Action potentials generated by autorhythmic cells create waves of depolarization that spread to contractile cells via gap junctions. Autorhythmic cells begin depolarizing due to a slow, continuous influx of sodium and reduced efflux of potassium. As sodium ions enter the cell, the inner surface of the plasma membrane gradually becomes less negative, generating the pacemaker potential. When the membrane potential gets to negative 40 millivolts, it has reached the threshold for initiating an action potential. Fast calcium channels open and positively charged calcium ions rush into the cell. Notice on the graph that the calcium influx produces the rapidly rising phase of the action potential depolarization, which results in the reversal of the membrane potential from negative to positive inside the cell. This reversal of membrane potential triggers the opening of potassium channels, resulting in potassium rapidly leaving the cell. Let's observe the potassium efflux and the resulting repolarization. The potassium efflux produces repolarization, bringing the membrane potential back down to its resting level. Although not shown here, several other events take place during repolarization. Ionic pumps actively transport calcium back to the extracellular space, while sodium-potassium pumps transport sodium out of the cell and bring potassium into the cell, thereby restoring ion concentrations to their resting levels. During the depolarization in adjacent autorhythmic cells or contractile cells, a few positive ions move through the gap junctions into neighboring contractile cells. This entry of positive ions brings the membrane potential to threshold, triggering voltage-gated channels and initiates depolarization.
So the only thing I would add to that is they implied that the calcium ATPase pump and sodium potassium ATPase pumps only work during repolarization, but of course we know they work constantly throughout the entire action potential and cardiac cycle. So now that we have a handle on the autorhythmic cells, I'd like to talk next about the contractile cells and the mechanism of how they get activated to reach threshold and how their action potential takes place. So let's draw another picture of the heart with its 4 chambers. Right atrium, right ventricle, left ventricle. Let's draw the SA node in again up here, and I'll pull it out and show you it enlarged with, of course, all of the cells connected by gap junctions. And these cells I'm drawing here are autorhythmic cells, and there would be a whole bunch of them all within this structure that I'm going to outline here and call the SA node. So this picture is just like the picture from the last lecture. Only now we're going to talk about what happens when that spreading wave of depolarization, that action potential, emerges in the periphery here of the SA node. Well, it turns out that some of these autorhythmic cells form gap junctions with contractile cells. So I'll draw one here. And those contractile cells in turn have gap junctions that connect them to other contractile cells, like so. So what we need to figure out next is how does the activation signal, the action potential that's traveling through the SA node, bleed out into the contractile cells and then spread in all directions as indicated by my squiggly arrows, from the right atrium over to the left atrium, to the rest of the right atrium, and all the way down ultimately to the AV node. So that's what we're going to talk about next. So as was shown on the video animation you guys just saw, the thing that actually causes activation of the contractile cells from the neighboring autorhythmic cells is the fact that there are gap junctions there. So I'm drawing this a little bit enlarged now, and we'll make a simplified drawing here of a contractile cell. So here's the SA node cell over here. Here's the contractile cell here. And we just saw that the action potential traveling across the membrane of the SA node results in an influx of calcium ions and sodium ions through various gates, channels that we've talked about, and that those ions then diffuse from higher concentration to lower concentration through the gap junctions. And it's that diffusion through the gap junctions that brings this contractile cell up to threshold. So diffusion, this is simple diffusion, let me be clear about that, simple diffusion of sodium and calcium ions through those gap junctions. Through those gap junctions, that's what brings the contractile cell up to its threshold so that it will generate an action potential. Okay? And we'll talk about the steps of that action potential in just a moment. But this brings the contractile cell up to threshold potential, which we'll learn is minus 70 millivolts for those cells. So up to threshold potential. And as a result, an action potential begins in the contractile cell. So there's only a split second between the action potential traveling through the last part of this SA node cell and a new action potential generated over the cell membrane of this contractile cell. So we get this propagation of the action potential with the little squiggly black line is my representation of that. From SA node cell down to contractile cell, and by the way, to the next contractile cell and the next, because they're all connected by gap junctions and they all work as a functional syncytium, as we talked about in a previous segment. So before we get into the details of how this action potential works in this contractile cell, I want you to take a quick look at slide 24. We've seen this slide before. This is the slide showing a longitudinal section through the heart, and highlighted in yellow are all the components of that intrinsic conduction system, or cardiac conduction system, the heart's little private nervous system, which consists of the autorhythmic cells, clumped together in the SA node at the top, in the AV node just further down, and through the bundle of His bundle branches and Purkinje fibers. But what's not shown on slide 24 is that all of those autorhythmic cells in there are connected by these gap junctions to these contractile cells. So I just want you to be aware, again, as the signal, the action potential depolarization wave moves through this intrinsic conduction system, it also bleeds out to all of the contractile cells by virtue of these gap junctions. And also, as a quick reminder of what these contractile cells look like, check out slide number 25. A cutaway view of a contractile cell. We'll talk about all the organelles inside and what they do, but I just wanted to remind you that the bulk of the volume of those cells is contraction apparatus, sarcomeres with actin and myosin and so forth. But so far, we first have to understand how the action potential happens across the surface of these contractile cells to then later understand how that in turn triggers a contraction inside the cell. And with regard to the action potential in contractile cells, I'd like you to take a look next at slide number 26. And that's a graph that shows the contractile cell's action potential in a blue graph. And then they also have another line there in sort of purple demonstrating the actual contraction process and when does maximum tension get developed. And so that you can see how that event of contraction is timed with the event of the action potential. So what's nice about this is that the events that happen in a contractile cell during an action potential are set up by the same preconditions that we learned about with the autorhythmic cells. These are your notes from the last lecture segment. I drew an SA node cell enlarged, and we talked about how these cells have some active transport pumps. The solid black circles are the sodium-potassium ATPase pumps, and we said they're pumping 3 sodiums out for 2 potassiums in, and there were calcium ATPase pumps pumping calcium out. Well, guess what? Those same pumps exist in the contractile cells. We talked about some voltage-regulated gates, calcium gates, potassium gates, and a sodium leakage channel. We're going to see in the contractile cell that there are gates, voltage-regulated gates, that dictate the events of the action potential. So let's take a look at a contractile cell, and let's draw in some of those components we've been talking about. One large contractile cell, looking at it close up. So of course we have those sodium-potassium ATPase pumps. I'll again draw them as black solid circles, and they're throughout this whole cell membrane. And again, what do? They pump 3 sodium ions out for every 2 potassium ions that get pumped in. And as a result of that, they create a gradient across the cell membrane, whereby sodium is more concentrated outside cells than inside, and potassium is more concentrated inside cells than outside. And then, of course, there's also the calcium ATPase pumps. Again, I'll draw them as open black circles, like so, and they are pumping calcium ions out of the cell, and as a result of those pumps, we have a concentration gradient for calcium, whereby calcium ions are more concentrated outside cells than inside cells. So we have the necessary concentration gradients having been built up already by active transport. These are sodium, potassium ATPase pumps and calcium ATPase pumps. They spend a lot of ATP energy to do this because it's active transport. It's pumping these ions against their concentration gradients. So my little, again, my black circle filled in here is the sodium potassium ATPase pump. And the open circle that I drew there is the calcium ATPase pump. The heart uses a lot of energy, a lot of ATP from cell respiration to create and maintain these gradients throughout an entire action potential, not just during repolarization or depolarization, but these pumps are working constantly, bailing these ions back out up their gradients. So we have the necessary preconditions for an action potential to occur. And by the way, it's because of these pumps that the interior, when this cell is at rest, is slightly negative with regard to the exterior. So when this cell is at rest, there's a resting membrane potential that's in the negative zone. I'm indicating this by negative signs on the inside and positive signs on the outside, just as we saw with the autorhythmic cells, our SA node cells. We have the same setup. And so if you had a galvanometer, again, a little probe that you stuck into the cell and compared the charge on the inside with the charge from a reference electrode on the outside, we call that a voltmeter. We talked about it earlier, same sort of device. And if you set up that device and took a reading when this cell was at rest, you would find that the cell membrane potential, CMP, measured in millivolts, when this cell is at rest, is a little further down than the autorhythmic cells. It's about minus 90 millivolts. If we put here time on the x-axis in milliseconds, so maybe 0 milliseconds, 100 milliseconds, 200 milliseconds, 300 milliseconds on the x-axis. So we have a resting membrane potential that's negative again. So we call that the negative resting membrane potential. I'll label that here. It's a negative resting membrane potential. Negative because it's relatively more negative on the inside. Resting because the cell is at rest, it's not undergoing an action potential yet. Membrane because the difference in charge, the potential, is across the membrane. And the word potential because it represents potential energy. So we have a negative resting membrane potential of about minus 90 millivolts. Minus 90 millivolts. These cells also have, as I said, voltage regulated gates for sodium, potassium, and calcium ions, similar to what we saw with the autorhythmic cells. So these are proteins spanning the cell membrane that can undergo conformational changes and either open up a channel or close a channel. So like a gate, they could be opened or closed. I'm going to draw in blue here the voltage regulated calcium gates in a closed position. And so my little blue arrow bar, if it's open, it might look like this, is the voltage regulated calcium gates. Voltage regulated calcium gates. By the way, in these cells, they're sometimes called slow calcium channels because when they do open up, they open up a little bit slower in a more delayed fashion. So the phrase slow calcium channels is sometimes used for these gates. And we also have voltage regulated potassium gates, which like before, I'm going to show in red, like we did before. And I'm drawing them as little red bars, sort of like a door that's in a closed position just for the sake of the drawing. And if they're open, they might look like this, like a swinging door that just opened. And these are the voltage regulated potassium channels or gates, like we saw similar to the other cells, the autorhythmic cells. And then with regard to sodium, it's a little different than we saw in the autorhythmic cells. In the autorhythmic cells, we saw there was sort of leakage sodium channels that were sort of like a door always propped slightly open. Well, in the contractile cells, we have voltage regulated sodium gates. I'm going to draw them as green bars here in a closed position, located all the way throughout in an open position like a swinging door. They might open like so. And those are the voltage regulated sodium gates. And because they open fairly explosively and quickly, they're sometimes called the fast sodium channels. So you'll see that phrase, fast sodium channels. And those open pretty quickly when a certain threshold potential is reached. And in these cells, the threshold potential is about minus 70 millivolts. So I'm going to draw a dotted line here just to let you know that that's the threshold potential for the opening of these fast sodium channels. So we call that the threshold potential. Remember, in autorhythmic cells, the threshold potential was minus 40. Here it's minus 70. So the numbers are a little different, but the concepts are similar. Now you might ask, how did we get from a resting membrane potential of minus 90 up to minus 70? How do we get from here on the graph up to this threshold level? Well, the answer is in these gap junctions and through these gap junctions, from the preceding cell, come diffusing in sodium and calcium ions from the action potentials of the next cell over. And remember, those ions diffuse by simple diffusion through the cytoplasm, through the gap junctions, into this next area here. And that is exactly what brings these contractile cells from minus 90 to minus 70. And that is exactly when these fast sodium channels swing open. And they're going to cause quite a sharp depolarization up above 0 millivolts, all the way up to about plus 25 millivolts, as you can see on slide 26, a sharp rise there above 0, up to maybe plus 20, plus 25-ish millivolts. And that's the upstroke of the action potential. Now, the action potential shape is going to look very different in these cells. It's going to quickly repolarize a little bit and then plateau out in a flat line for a while, and then finally repolarize all the way back down to rest. So this shape of this AP, or action potential, is a little different than the shape we saw in the autorhythmic cell. But the general concepts are the same. Action potential would not be possible were it not for the previous setting up of these steep ion gradients. And in the same way that we saw with the autorhythmic cells, because of these ion gradients and the concentration gradients and the electrical difference in charge across the membrane, each of these ions has a certain equilibrium potential that if allowed to, if this cell were only allowing a particular ion, like perhaps calcium ions, to flow through the membrane, if we could rig this so that only the calcium gates were open, calcium would rush in down its electrochemical gradient, and the cell membrane would strive to reach calcium's equilibrium potential, which, remember, we said is way up above 0. I think we said about 130 plus 132 millivolts. So during this action potential, whenever I say that a calcium gate's going to open, you should immediately think that's going to depolarize the cell membrane, because the cell membrane is going to strive to reach calcium's equilibrium potential. The same with sodium. Sodium's a positively charged ion, more concentrated outside than inside. If we were to just open up a sodium gate, like these fast sodium channels, look what happens. We sharply rise. Why do we rise? Because sodium ions are rushing in, bringing their positive charge into the cell, striving to reach that equilibrium potential for sodium ions, which remember we said was about plus 70 millivolts. And then even potassium ions, they're more concentrated on the inside than the outside. So if ever we say, here's now when the potassium gates open, you should conclude potassium's generally going to rush out of the cell, take positive charge out with it, make the interior more negative, and cause us to repolarize back down. And in fact, this steep cliff downward here on the end of this action potential is exactly because of potassium gates opening. What is potassium doing? It's striving to reach potassium's equilibrium potential, which we said earlier was minus 94 millivolts. Now notice, any rise in the cell membrane potential suggests either sodium and or calcium permeability, and any fall in the cell membrane potential back to rest suggests an increase in potassium permeability. But notice, we never quite reach the equilibrium potential. We get nowhere near plus 70, sodium's equilibrium potential, or plus 132, calcium's equilibrium potential. And on the way back down, we only get to minus 90. We don't get all the way down to minus 94. So what does that suggest? That suggests that something is stopping us from reaching that equilibrium point. And it's always the closure of gates. So the same gates that open up and cause depolarization are actually designed to close before we reach that settling out equilibrium potential way up at plus 70 or plus 132 millivolts. Similarly, when the potassium gates open and we repolarize back down, we never reach minus 94 because those same potassium gates that opened to cause us to fall quickly here are going to close the further down we get. So it's like the cell membrane striving to reach calcium and potassium's equilibrium potential, doesn't quite get there, and then other gates open and we strive to reach potassium's equilibrium potential, doesn't quite get there, and then other gates bring us back up. So it's an alternating depolarization and repolarization. Really, this concept is sort of the same as we saw with the autorhythmic cells. So let's now go through the steps of an action potential as it happens in a contractile cell using all of the principles and concepts that we've been talking about. So, and let's start out with a cell that's at resting membrane potential, nothing going on, and we'll call this steps in the contractile cell's action potential. Steps in the contractile cell's action potential. AP is my shorthand, as you know, for action potential. Okay? So to start out, the cell's at rest. And that cell will move from resting membrane potential up to, which is minus 90 millivolts, up to minus 70 millivolts because of calcium and sodium influx from neighboring cells. So you've got sodium ions and calcium ion influx through gap junctions. JXN is my shorthand for junctions. So you got sodium and calcium ion influx through gap junctions brings the cell membrane potential from minus 90 to minus 70 millivolts. So from that resting state of minus 90 millivolts up to minus 70 millivolts. Then we call that the threshold potential, because that's the threshold that has to be reached for those fast sodium channels to open up. So that's exactly what happens next is step 2, those voltage regulated sodium gates or fast sodium channels, as I described them, open up quickly. And when they open up quickly, fast, they allow an inrush of sodium ions. So allowing sodium ions to rush in by facilitated diffusion, as you know, into the cell. And that's happening by facilitated diffusion, through those fast sodium channels. And when that sodium rushes in, sodium ions rush in, they're bringing positive charge. So this causes sharp depolarization, a sharp upstroke of depolarization in our graph of the cell membrane potential. And that sharp upstroke of depolarization brings the cell membrane potential up to 0 and even beyond, up to about plus 25 millivolts. The numbers vary from cell to cell, but approximately up to about plus 25 millivolts. All right? And that's called the upstroke of the action potential. And if you again look at slide number 26, our graph of the action potential, the blue line, remember, look at how steep that upstroke is. It's an explosive opening of lots and lots of sodium gates. And that's where the opening is so quickly, it causes such a sharp depolarization. These are called fast sodium channels. But those fast sodium channels, they're voltage regulated. And just as they're changing shape to open at minus 70 millivolts, they change their shape again to close almost as quickly up at plus 25 millivolts. So at, again, 25, 30, it varies from cell to cell. We'll just say at approximately plus 25 millivolts, those voltage-regulated sodium gates close again. So they don't stay open for very long. Those gates close again. This is why we only go up to about plus 25 rather than going all the way up to plus 70, the equilibrium potential for sodium ions. But those voltage-regulated sodium gates close again, stopping further depolarization of the cell membrane potential. The other thing that keeps this cell membrane potential from going all the way up to sodium's equilibrium potential is also at about plus 25, a small number, a small percentage of the voltage-regulated potassium gates start opening. So at approximately plus 25 millivolts, a small percentage of the voltage-regulated potassium gates open up. Not all of them, a small number of them, but those voltage-regulated potassium gates open up. And of course, that allows potassium to rush out of the cell, making the inside of the cell more negative. And that's why we go sharply up, but then we take a sharp turnaround in the graph. So again, if we graphed this, we have resting membrane potential and then a sharp upstroke, but then a very quick turnaround. And actually a sharp turnaround, not all the way back down to minus 90 millivolts, cell membrane potential in millivolts, but down a little bit, and then there's a plateau phase where things flatten out before we repolarize back down to the resting state. So we don't get all the way up to plus 70 millivolts because those same sodium gates that opened at minus 70 close once you get up above 0. And also because once you get up above 0, a small percentage of the voltage-regulated potassium gates open up. Now, what I didn't tell you is there's actually more than one sub-variety of voltage-regulated potassium gates. Your book doesn't really go into that. I'm not going to expect you to know that. But just realize that a certain variety of them starts to open, and that's why we have this about face turnaround, and instead of going straight up, we briefly repolarize onto this next phase, which is the plateau phase. So the next phase of this, I'll number this number 5, we're going to call the plateau phase. Plateau means flat, so that plateau phase, which I'm going to label right here. So let's try to understand, why is everything flattening out at that point? Okay? There's a few things happening during the plateau phase. As I said, some voltage regulated potassium gates have opened up, and that has allowed some potassium to leak out of the cell. So there is some potassium ions leaking out of or diffusing out of the cell by facilitated diffusion, diffusing out of the cell. But the amount of positive charge in the form of potassium ions moving out, turns out it's going to be equally matched by the amount of positive charge due to calcium ions moving in. So there's some potassium ions diffusing out of the cell is equally matched in terms of charge by calcium ions diffusing into the cell. Okay, so in other words, the amount of positive charge moving out in the form of potassium ions is equal to the amount of positive charge moving in by calcium ions. Now, we haven't talked yet about calcium ions, but why are calcium ions now rushing in to the cell? Well, it turns out that on the way up in this sharp upstroke, hidden in this sharp rise earlier that was due to sodium ions rushing in, at about minus 30 millivolts, there's another threshold. And it's a threshold potential for the calcium ions. So whereas minus 70, we said, is the threshold potential for the sodium ions, for those sodium gates, at minus 30 is the point when voltage-regulated calcium gates open up. Voltage-regulated calcium gates open on the way up at about minus 30 millivolts. That's at minus 30 millivolts, whereas the threshold potential for the sodium gates was minus 70 millivolts. So it turns out that the last part of this upstroke is not only due to sodium rushing in, but to calcium rushing in. And calcium gates, they're not as fast as the sodium gates. They're sometimes called slow calcium channels because they, as a population, they open up more sluggishly, more slowly. But by the time we reach this this plateau phase, there's enough calcium moving in through those channels to match the charge in the form of potassium moving out. So this plateau phase represents no net change in the cell membrane potential because the amount of positive charge moving in is equal to the amount of positive charge moving out. So just to complete the notes here, the amount of positive charge diffusing out in the form of potassium is matched by the amount of positive charge flowing in the form of calcium ions. And I'm just going to put a brackets here. Those calcium ions are diffusing in. They're moving in through voltage-regulated calcium channels, or calcium gates. And as I mentioned earlier, these are sometimes called slow calcium channels to contrast them with the fast sodium channels. And when I say slow and fast, I'm actually talking about two specific things. How fast does a particular gate open? And as a population of, say, 100 gates, how fast do we reach the point where all 100% of the gates are open? So in the terms of sodium gates, each gate opens up quickly, and as a population, they all open up quickly. But in terms of the calcium gates, each gate is like a door that opens more slowly, and as a population of them, it takes a little longer for all 100% of those gates to open up slowly. So really, for two reasons, we call these slow calcium channels. But anyway, we have a plateau phase. Of course, that plateau phase doesn't last forever. Eventually, we undergo the last step here, which I'm going to call repolarization of the cell membrane. And that would be when we're looking at a graph of all of this, and we've been through the upstroke, the plateau phase. Now we're talking about this return back to resting state, this repolarization. And that is an instance where the cell membrane potential is getting closer down to potassium's equilibrium potential, which is minus 94 millivolts. So if you were to just wager a guess as to how is it that we go from the plateau down to minus 90, we almost get all the way to minus 94, you'd have to say it must be because the cell membrane is becoming more and more permeable to potassium and less permeable to the calcium. So at the end of the plateau, we see these changes in permeability of these two gates. So I'll just write this down. At the end of plateau phase, the slow calcium gates, the voltage-regulated calcium gates, start to close. They start to close as more and more voltage-regulated potassium gates open. As more and more voltage-regulated potassium gates open. So now the cell membrane is becoming essentially just permeable to potassium. So what does it do? The cell membrane potential strives to reach potassium's equilibrium point, which we know is minus 94. And so at the end of the plateau, voltage-regulated calcium gates close as more and more voltage-regulated potassium gates open, allowing more potassium to diffuse out of the cell. And as more potassium diffuses out, this is going to repolarize the cell membrane back to our resting state of minus 90 millivolts. This is going to be repolarizing the cell back to minus 90 millivolts. And where are we then? We are at the start of the next cycle. So we've just undergone a complete action potential marked by resting membrane potential, sharp upstroke, plateau phase, and repolarization. Now, I will stop there, but in the next segment, we'll talk about how that action potential not only spreads over the surface of the contractile cells, but also what is it about that action potential that then actually triggers a contraction, a mechanical shortening of those contractile cells. And we'll get to that at the next segment. So we'll see you soon.